Today we will be discussing on the genetic testing options available and which testing options should we go for in which cases. All right, so let's start with the presentation. So as I mentioned, I work at Centogene, which is a global leader in the field of genetic diagnostic testing for rare genetic disorders. Centogene's mission is to support medical professionals with in-depth medical expertise to diagnose early and safely the reason for patients' burden. This is our global footprint. We are present in more than 120 countries and we collect samples from more than 35,000 physicians. So before going into knowing more on genetic testing, let's dive a bit into the human body. So we know that the human body is made up of millions of cells. Each cell has a nucleus inside it, right? And inside the nucleus, humans have 46 chromosomes. That means 23 pairs of chromosomes, 22 pairs of autosomes, and one pair of sex chromosomes. Correct. And if we zoom in more into the chromosomes, we will find long stretches of DNA known as genes. Correct, which is the protein coding unit. And when we focus more into the DNA, it is made up of single nucleotides, right, which codes for the proteins that the body needs for the structure and function. Now let's focus a bit on the contents of a gene. So each gene is made up of a promoter region, correct? Then the upstream regions, we have the 3' upstream region and the 5' upstream region, and it is made up of exons and introns. Now exons are actually the protein coding regions which are finally transcribed into the mRNA, and the mRNA translates into a protein which is actually does all the function in our body, right? Now what if there is a change or there is a mutation in the DNA, right? So that leads to genetic disorders that we know of. And what are the type of changes that can happen in the DNA? So starting from the smallest change, that means it is an SNV or a single nucleotide variation, right? Means a single nucleotide in the DNA is changed. It can be an insertion, it can be a deletion or a substitution, correct? So here it is shown that here G is C nucleotide is substituted to T, correct? So one classic example of a disease caused by a single substitution we know of is sickle cell anemia, right? So in beta globin gene in exon 6, a single substitution event leads to an abnormal hemoglobin, right? Which then causes a sickle type shape of the hemoglobin, correct? And which leads later to anemia. And if we go a bit into larger changes, that can be an insertion or deletions of more than one nucleotides, correct? A classic example of a deletion of 3 bases or 3 nucleotides is in the CFTR gene, correct? Resulting in cystic fibrosis. A 3 base pair deletion in that gene results into an abnormal protein, correct? And it loses its chloride channel activity and then it leads to excess of thick mucus production, which then leads to lung dysfunctions or pneumonia, right? Then coming to a bit of a larger change, that can be a larger deletion, right, which is constituting of more number of nucleotides, or a duplication in which a particular DNA region is duplicated, correct, or an inversion, right, or inversion of the DNA sequence. or an extra insertion of a DNA sequence, right? Or a translocation means the exchange of DNA fragments. These are known as structural variations. Some classic examples are Duchenne muscular dystrophy, which is caused by either deletions or duplications of certain exons in the DMD gene. Correct. Then a classic example of translocation is Philadelphia chromosome, right? Which leads to leukemias. Then for inversion, there is a classic example that if the intron 22 or intron 1 of the F8 region is inverted, right, it leads to hemophilia A. Correct. Then these are structural variations which are larger than a single nucleotide variations constituting of multiple number of nucleotides. Then coming on to a bit of a larger change, correct? These are chromosomal changes. That means the entire chromosomes have been altered. In some cases, it may be an addition or a deletion of a single chromosome out of the 46 chromosomes. Correct. So if there is an addition, right, it is known as trisomy. So in normal humans, there are two pairs of each chromosome, two of each chromosome, that is one pair. But in trisomy, there is one additional chromosome. Correct. So a classic example of this is Down syndrome, right? Due to an additional chromosome 21, trisomy 21, right? Then there can be a deletion of a complete chromosome called as monosomy. We know of a classic example, that is Turner syndrome, where there is only one copy of a sex chromosome, that is there is only one X chromosome, the other sex chromosome is missing. Or there can be large scale gains, that is the complete chromosome ploidy is changed. We are diploids, right? We have two of each chromosome, but sometimes in cases where there are three of each chromosomes, that is the complete ploidy has been changed. We know that this is possible when there is a triploidy, correct? That means one additional set of each chromosome is present. Then there are certain changes in the DNA known as repeat expansions, which can also result into a disease. That means in normal individuals, these repeats, these can be a trinucleotide repeat or a pentanucleotide repeat. These repeats are present in the genes, correct? But sometimes, mainly in the spermatogenesis or oogenesis process, these repeats expand, right? And once these expand, once these pass a threshold, then this causes disease. The examples for repeat expansion disorders are Fragile X syndrome or spinocerebellar ataxias, correct, or Huntington's disease, right? Then some of the mitochondrial genome variations can also lead to some diseases, right? We know of Leigh syndrome, which causes neurological disorders, that is mostly related to changes in the mitochondrial DNA. Then there is one more DNA change called mosaicism, but which is not present in all cells of the body. In the process of embryogenesis, maybe one or some cells of the embryo gain a mutation, and then only the cells which are arising from those mutated cells have the mutation. That is known as mosaicism. So that particular DNA change will be present in only some cells of the body. The other cells of the body will be normal, right? Creating a mosaic pattern. And other than DNA changes, there are certain changes other than the DNA, right, known as epigenetic changes, which also lead to disease manifestations, right? These are mainly DNA methylation patterns, right, mainly on the promoter regions of the genes, which can either suppress the genes, correct, or which can either overexpress the genes, right? As we see in some imprinting disorders, correct, Prader-Willi or Angelman syndrome or this also, epigenetic changes also play a huge role in cancers, right? So now we know that what are the DNA changes that can happen inside the body. Now let's see that under what cases which genetic tests should we go for, right? So in cases where the patients have large scale chromosomal changes, right? And how would we know that which tests to go for that for these changes, for these large scale chromosomal changes like monosomies or trisomies or complete polyploidies, right? These large scale chromosomal changes, there are certain classic phenotypes which are present in the patient, right? The patient would have mild to severe cognitive impairment, the patient will have delayed milestones, abnormal growth patterns, mainly congenital malformations of the heart, kidney, and brain, facial dysmorphisms, skeletal changes, microcephaly or macrocephaly, and skeletal abnormalities. And in these phenotypes, if the patient has been identified with these phenotypes, one thing to check is that whether is this hereditary. Correct, because most of the chromosomal changes are not hereditary. And why? Because they take place in the process of oogenesis or spermatogenesis, mainly because of non-disjunction of the chromosomes, or sometimes also during the process of embryogenesis. So these uh, phenotypes or these chromosomal changes cannot be inherited, right? So if you see a hereditary pattern in the family, right? So checking for large chromosomal changes wouldn't be a good option. Correct, because most of the large chromosomal changes are de novo. Correct, that means they are not in the parents but in the children. One exception is the Robertsonian translocation, right, which can be passed down from the parents. Other than that, all are all arise only in the baby. Okay, so in these cases, for large chromosomal changes, when you identify these classic phenotypes, correct, or when you see that the pattern is not hereditary, then the best test to go for is either a karyotyping or a chromosomal microarray, which can detect these large chromosomal changes like trisomies or monosomies or polyploidies. Right. Now, for patients, correct. If you have a classical phenotype completely correlating to a disease, right, and if that disease is caused by only one gene, right, so in those cases, it is better to go for single gene tests, correct. Like like example of cystic fibrosis, the phenotypes are for cystic fibrosis are classical for cystic fibrosis, like chronic lung infections, right, or progressive lung damage and pneumonia, then pancreatic insufficiency, and then intestinal blockages, especially in the newborns, elevated chloride levels in the sweat, then failure to thrive, right? And for these classical symptoms, you know that it is cystic fibrosis, and you know that there is only one gene causing cystic fibrosis. And in this case, you also have to check for the inheritance pattern. The inheritance pattern for cystic fibrosis is autosomal recessive. So you also check the inheritance pattern. If it is discordant with the inheritance pattern observed in that family, then maybe going for this particular single gene test is not a right option. So in these examples, where the patient is presented with some classical phenotypes for a particular disease, and you know that for that disease, only one gene is responsible, correct? And the inheritance pattern also matches. Then the best is to go for single gene tests. One more example is for neurofibromatosis, right? These are the classical phenotypes, multiple Caffioli spots, then neurofibromas, Lish nodules, right? Then there are certain learning disabilities, scoliosis, correct? In these, here you would know that it is neurofibromatosis and only one gene is mainly responsible for neurofibromatosis, and the inheritance pattern in the family is also consistent with an autosomal dominant inheritance pattern, right? Then you go for a single gene test of NF1 next generation sequencing, right? But in cases, right, where the symptoms for that patient are not suggestive of one disease but lie into a single category of disorders, maybe cardiovascular disorders, or maybe neurological disorders, or nephrological. So, in that cases, the best test to go for is the panel testing, right? These are all the panels that Centogenes offer. So, in cases where you see a patient where most of his phenotypes, correct, belong to a particular category, right? It can be hematological, it can be immunological, then the best is to go for a panel testing. Now, here the panels will include all the genes responsible for that particular category, right? So you would be sure that if the patient has cardiovascular symptoms, all the genes related to cardiovascular diseases or symptoms or phenotypes would be covered in that panel, right? But in the cases where the phenotypes don't fall into one single category, right? The phenotype is complex, involving more than two categories. That means cardiovascular together with neurological, right? Or another combination. Then in such cases, it is better to go for whole exome sequencing. That means sequencing of the exonic regions of all the genes, correct, in the human body. And also, if you're suspecting a mitochondrial disorder, then this test is a better option. And also, if the karyotyping or CMA test, you have suspected a large chromosomal change, but the karyotyping or chromosomal microarray has failed, then in these cases, the best genetic testing option to go for is the whole exome sequencing. Now, at Centogene, these are the product specificities that we offer in whole exome. We cover the entire exome, also along with the splice site regions and also the mitochondrial genes, right? Our coverage for more than 98% of the region is more than 20x. And we give an enhanced coverage for the variants that are already reported in OMIM or NGMD. And we have a higher sensitivity for copy number variations as well, right? We also detect mitochondrial heteroplasmy as low as 15%. And we have also included uniparental disomy detection for all known clinically relevant regions. We are also able to detect mosaicism in a whole exome sequencing and also imprinting disorders. Right. Some of the advantages here in opting for our whole exome sequencing is that we give a higher data output. That means we generate more data for each patient. Our copy number variation sensitivity is also high. So an additional ML, if you get a negative report, then the dependency of an additional MLPA test is reduced because our copy number variation sensitivity is high. We detect known uniparental disomy regions. In our whole exome, the mitochondrial genes are also included. And if in case we have identified a variation in a low coverage region, for suppose, we do a Sanger validation by ourselves. It is inbuilt, right? And we also do the validations for copy number variations by orthogonal methods like MLPA or qPCR or chromosomal microarray, right? Our VUS percentages, that is percentage of variants of unknown significance, is very low. And even if we get a VUS, we provide a lifelong diagnostic support that every 3 months our team continuously re-evaluates those variants of uncertain significance, and if more additional data is available and if we have reclassified that VUS maybe into pathogenic or likely pathogenic or maybe into benign or likely benign, we re-release the report of that patient even if it is 2 years older or 3 years older, we re-release the report. We inform the physician that, hey, 3 years back, you had given us a patient in which we had reported a variant of uncertain significance. But because of new evidence, we have reclassified that variant as pathogenic, and we are re-releasing the report saying that now this variation is pathogenic, right? This helps to solve many of the dilemmas which is created by variants of uncertain significance, right? It helps solve patient's anxiety as well, that finally the patient has a positive or a negative report, correct? And now in cases, for suppose you have given tests, the previous genetic tests, and all have come back as negative. Then there is one last resort that you can go to whole genome sequencing, in which the entire DNA of the individual is analyzed along with the intronic region, along with the promoter regions, correct? And it is noted that 30% of the cases which come negative with whole exome sequencing come as positive in whole genome sequencing. Right? So now I mentioned about VUS, right? Variant of uncertain significance. Now let's learn a bit more on how actually the variants are classified. So there are 5 categories, right? Starting from benign, likely benign, then variant of uncertain significance, likely pathogenic, and pathogenic. And a variant, according to ACMG, can be classified in either of these 5 categories, correct? And based on what? So if that variant, right, the disease causing probability of that variant is more than 99%, it will be known as pathogenic. If the disease causing probability is more than 90%, it will be known as likely pathogenic. If the disease causing probability is between 10 to 90%, then it is variant of uncertain significance. And likewise for likely benign and benign, if the variant or the disease causing probability is less than 10 or 1% respectively, then the variant will be classified as likely benign or benign. So what is a positive report? A positive report is when in that patient we have identified either a VUS or a likely pathogenic or a pathogenic variation, right? And once we have identified the variation, we also go through these medical evaluation steps. That is, once we have identified a variation, first we check that what is the phenotype overlap, correct? What is the phenotype overlap caused by the gene in which the variation is obtained and the phenotype of the patient, correct? Is that overlap partial? Is that full? Is that none? Correct. We also look into mode of inheritance. So, for example, a gene is known to cause an autosomal recessive disease, and we obtain a heterozygous variation in that gene, correct? That means the mode of inheritance is not matching, right? Then we will look for more variations. We also match the age of onset for the manifestations known for that gene and compared to the age of onset of the patient. We check for family segregation if the inheritance pattern is segregating with the family. We also look into the disease mechanism, whether is it a gain of function or a loss of function, correct? So these are all the medical evaluation steps we take into consideration once we have identified a variation for an individual. So this is the process that we follow. So initially, for a patient, we take the blood on a filter card, which we call as Cento card. Here, we can extract DNA from this card for more than 10 years. So in case resampling is required for another test for an individual, correct? Or re-isolation of the DNA is required, we already have the blood sample. We don't have to resample the blood from the patient, right? And once we collect the sample on the Cento card, we ship it to Germany, right? And for the patient's clinical details, we take all the clinical details, all the medical reports from the physicians and from the patient, correct? Then we enter those details into Cento portal. And simultaneously, the blood sample goes through next generation sequencing, goes through the bioinformatics pipeline, and finally, the medical evaluation team evaluates the variations obtained in the patients with the clinical information provided. Right? They check that whether the variations, the phenotypes are overlapping, correct? Or whether the mode of inheritance is correct, is it segregating with the known family history, the age of onset is matching. Medical evaluation teams checks everything and finally releases the report. So this is the complete process of how we identify a DNA alteration in a patient, correct? And I've gone through options to go for DNA or genetic testing options to go for in case of a particular, uh, in case of phenotypes, that whether they are classical, whether they are complex, whether they are falling into a particular category, whether the inheritance pattern is matching, correct? So this is all. So if you have any questions, please put in the chat.